Thank you very much. It's so great to be here in person again after this long COVID period. And I want to, I'm the one who tried to use aptamers to control gene expression, to develop tools. And we hope we develop them so well that other groups from basic science then may use and apply them. Okay, and so in the first part, I will tell you some old stories, and in the end, I will present our new kit. So, okay, always start with here this my most favorite textbook picture that shows that my most favorite and beloved RNA molecule is able to adapt different conformations. So it's a flexible molecule, and nature is using this to control gene expression. This is known. These are the ribose switches. So they use this alternative conformation. They are highly structured RNAs located in the 5' UTR of many bacteria, mostly metabolic genes. They can have an aptamer domain, and the expression platform can sense a metabolite directly, and then the expression platform reads out the binding status of this aptamer and then either switch on and off gene expression. And why do we like them so? On the one side, there are, they are so small and defined. You do not need, like with the TET system, you do not need an additional protein to implement and to change the promoter and have some issues with the integration site in the genome. Then, of course, you have this high affinity and specificity. You have the RNA that can sense and regulate. Due to the modular nature, you even can construct complex switches, so you can play like Lego with them. And then, of course, we all know our select method we like. And I think here in the audience, I do not have to explain what this means. If you are lucky, then you can select really binding domains de novo. So, and we want to use these mostly small molecule binding aptamers, in the last time also protein binding aptamers, to develop switches that can be easily used to control gene expression. And how do we do this? So we want to develop them independent of the organism. So we try to develop them for yeast, for archaea, for human cell lines, sometimes also for bacteria. And we have different strategies. So the simplest one is shown here. Here, the aptamer itself is a kind of simply obstacle or roadblock. You put the aptamer in the 5' UTR, and then if the ligand comes, it gets stabilized and can interfere with the scanning ribosome. Or if you put it close to the ribosomal binding site, you can interfere with the binding of the ribosome. Similarly, you can interfere with splicing processes. Okay, this was too fast. Or you can, and this is also a possibility, use aptamers to control ribozymes. So these are this whole field of aptazyme development. So there are a lot of different strategies where you can try to control gene expression on the level of RNA. And here today, I only want to give you one example. So this again is our most favorite aptamer. It's a tetracycline aptamer. You see tetracycline in the middle in green, high affinity binding, 600 picomolar. And this was our very first experiment. I think it was around 2000 when I did this measurement. where you simply put an aptamer in front of the reported gene in yeast, and you can control gene expression in a dose-dependent way. In the meantime, we have optimized it. We used KI for optimization of the context. So these elements are highly context specific. You can increase them if you put more than one, and so on. And here, because this obstacle thing, it simply it works only in yeast. So then we thought a long time, how can we control also eukaryotic gene expression? And here, this is a strategy that in the meantime really works excellent. You can nicely switch on gene expression of your gene of interest if you work on the level of alternative splicing, if you try to control this. The idea is that you divide your mRNA into parts, or use natural introns of your gene of interest, and you insert a poisoned exon, yeah? And you control if this poisoned exon or this exon is included or not. If it's included, then you have here additional stop codons, so the message will be degraded by NMD. And if it's skipped, so if we have here this splicing decision, you end up with the correct message. And yeah, this works pretty good. And you simply have to take this part and you can put it into your gene of interest. You can either put it into a natural intron, or you can create an artificial intron and so on. And this works pretty nice. We already published it for CD20, but here brand new data of student in my lab, Robin. Here we try to control a key enzyme in the leukotriene biosynthesis. It's the 5-lipoxygenase. Again, here is the mRNA, the coding sequence in green, and this is a natural exon, an intron of the 5LO, and then we inserted our controlled exon, our poison exon, and here we did a lot of different studies. So what should we retake of the natural intron, and how long it should be, and so on. And finally, after a lot of optimization steps, and maybe you only focus here on these two lanes. This is a Western blot of the protein, and this is without tetracycline, and this is with tetracycline. And this is, I think, extremely impressive how massively you can switch on gene expression simply by adding tetracycline to your cell culture. So this was only as a kind of introduction to show you that indeed there is these are useful tools, and we hope that in the future many more people will take our devices and not only this classical TET-on TET-off system, or at least consider our regulation as an alternative. So yeah, what is the problem? The problem in the field, and I always talk about this, is that there are only a few aptamers that can be used for regulation. So theophylline, widely used. We like tetracycline, but we tested many of them. So there was a PhD student in my lab, and I think he tested 20, 25, and they all failed. So the most of the small molecule binding aptamers do not work in this way. And so one main focus of my lab is to understand why this works, and then to finding solution to easier find dysregulating aptamers. So big question for me to solve until my retirement: what turns an aptamer into a riboswitch? To do this, you have to compare inactive aptamers with active ones. So aptamers that can be used for regulation with them that bind well but do not regulate. And we started here with neomycin, and our way of finding these aptamers is the following. We think that selects alone is not sufficient because selects only for binding. And this is not what we are looking for. We're looking for aptamers that bind and regulate. And so we have always a selection and couple it to an in vivo screening. In this case, we like yeast. We have an enriched aptamer pool, put it in front of an aptamer, and then look which yeast show a ligand-dependent change in gene expression. Here with neomycin, we are lucky and we could find this little guy here. It regulated gene expression, simply comparable to the tetracycline ones, but here, if you sequence this pool, and at this time we did the classical Sanger sequencing, you do not find this candidate, you mostly find this one. And then we had two aptamers. One could regulate, the other not. The KD was the same. And then we thought, okay, we have to look into the structure. This was done by the Wernert group in Frankfurt. And they could show that here, indeed, we have a kind of structure switching that comes along with the ligand binding. So you have here the UU base pair forms, and this allows the formation of this GC, and then this GC stacks on this GC, and you have here this straight A form helix that is somehow stabilized by the ligand. This acts like a clamp here, yeah? So this was one major point, we think, or is one major point in making aptamers regulable. Here you can see the structure. Yeah. We went on another ligand, this time ciprofloxacin. We did exactly the same selects in vivo screening. We found, could find some aptamers, and we compared regulating with non-regulating ones here. This is one candidate here, KD 30 nanomolar, no regulation at all. Here, KD 60 nanomolar, nice regulation. Yeah. This time we had a closer look to the kinetics of binding, and what we could see here, this was a collaboration with the group of Sepp Wachtweidel in Frankfurt. They could show that the two subtype or the two aptamers have different binding kinetics. In both cases, we have a two-step binding, but our regulating aptamer, the second step is really tight, so you have nearly no back reaction from in the second step. And this was interesting for us because we thought, okay, if we make a classical Selex, and this is really such a specific binding kinetics, then our aptamer may stick to the column and we cannot elute them. And so we indeed could show this by deep sequencing. We have our normal aptamer that do not regulate. We have a 400-fold enrichment in the selection, but our value switch here, there was only a 15-fold enrichment. So this binding kinetics, the special kinetics somehow seems to, or in the classical selects, we lose our good candidates because they somehow stick to the column. And we could in the meantime show that this is not only true for the tetracycline aptamer, but we could see the same for the ciprofloxacin aptamer, we could see the same corresponding kinetics also for the tetracycline and the neomycin aptamer. So this was true for all of our three aptamers. Yeah, this I think are two important things, this two-step binding and the structural switching and the problems that arise in a classical Selex. We select for binding and not for switching. Ribo-switching aptamers, they stick to a column somehow, and for us also the immobilization of the small molecules is a nightmare, so we are not chemists. So. And then Adrian, he's here in the audience. When he joined my lab, he said, I will try capture Select. So I think Jean-Jacques Thomé told you should try this. And then there was no way to say no. He said, no, I will do it. And then he really had a hard time, but finally he got it running in the lab. And here we think this is how capture Select works. Here you immobilize not your ligand, but the pool via a small capture oligo, and then you have the pool immobilized here on the beads, and then you add the ligand in solution, and you elute only this aftermath that can somehow, no one knows really how, dissolve from this column, binds to your ligand, and then you can repeat this cycle again and again and amplify aftermaths that do not bind, not only bind, but also change their conformation. And on the other side, we do not have to immobilize our ligands, and our aftermaths do not stick to the column any longer. So this was the work of Adrian. He chose paromomycin, Select again, in vivo screening again, and you can here see this aftermath that regulates this paromomycin and the ligand specificity. This is really great. So paromomycin and neomycin, they differ here only in this one side chain. We have an OH for paromomycin, amino for neomycin, and our paromomycin aftermath, you can switch the trigger with paromomycin, but not this neomycin, and our old neomycin aftermath on the side can be controlled by neomycin, but not by paromomycin. So here only as an example how highly specific these aftermaths are. And then I like this plot, because here we try to find out, or we looked where we find our regulating aftermaths in our enriched aftermath or during the selection. So we had a look into the deep sequencing data, and the ciprofloxacin, this was one that was found by a classical Select. And here we could not find this precursor among the top 1,000. So the chance of finding it was 1 in 20,000. So it was really luck that my student could pick this candidate. Normally we screen 2,000 to 3,000 yeast cells, and here he was really lucky. The paromomycin aftermath, this was the one that was found by capture Select. This candidate we could find among the top 100, and the chance of finding was 1 in 900. So there's a much higher chance finding this. And of course, you cannot really compare apple with yes. So currently we try to select aftermaths against one ligand with classical Select and capture Select and do a real comparison. These are ongoing studies in the lab. But I always say, okay, now it's so easy to find new riboswitches or regulating aftermaths. And then, yeah, here are some new data to maybe to convince you that this is true. Again, a Select this time. Yeah, we started with kanamycin, was done by a guest and by Adrian in the lab, and then several students, and Leon, he's in the back here, he took over the project. And yeah, I think important are these small numbers here. Okay, he only, or we screened only 800 candidates, so really a small amount. And in the earlier time, we screened 3,000 and more. And we found 2 positive candidates, and you can see them here, and they both regulate gene expression. Yeah, they have a common motive, but we found it twice. In Violet here, you can see the capture sequence. And yeah, we of course, we optimized it. We shortened it. So first we deleted this, this head part. This resulted in T1 that regulated much better. And then to our surprise, we could switch this riboswitch not only with kanamycin, but also with topramycin. And it worked much better with topramycin than with kanamycin. You can see here, topramycin is the dark color, and here you have a really nice regulation of gene expression. You can switch it with CanR, CanB, and topramycin. Topramycin, it's not really surprising. It has one amino group more than kanamycin, so has maybe a higher affinity to the RNA. But so we decided we have now a nice topramycin riboswitch. Then here we did an ITC, and what we could see is this biphasic behavior. So this indicated two binding sites. And this, again, the group of Jens Wöhler in Frankfurt, they did some NMR experiments, and they could nicely titrate both binding sites. Here you see some more peaks popping up if you add more tetracycline, and he could then show that this first peaks correspond here to the lower part, and these additional peaks coming at a later or at higher concentration correspond to the other part. And then we said, okay, if we have two, maybe we can divide them or dissect them, or maybe there is even a cooperative binding. So yeah, we divided the aptamers into parts, and indeed they both bind topramycin, the lower binding site with 2 nanomolar, the upper binding site with 100 nanomolar. But when we tested them, then the lower binding site, the blue one, this is a real riboswitch, and the red one, it does not regulate. And then we also took the complete, and we deleted by point mutation the upper and the lower binding site, and we could show that there is no cooperative binding. So it's indeed only the lower binding site that has this riboswitch activity. And then we wanted to further improve them. We did a lot of mutagenesis. We somehow, there was an interesting effect. We again, we could not improve the KD of our aptamer by mutagenesis. So it seems that with the selection, we already achieved the best candidate by selects in terms of binding. So we never ever managed, and we did a lot of mutagenesis to improve the KD, but we could improve the switching, and the mutations we introduced were only in the peripheral elements. So kind of expression platform. So we decided that we may not need this upper part. So we cut it. We had an randomized N6 pool, and we screened, and we could here with this optimized apical loop further improve the regulation. But the binding behavior as itself does not change. So, and here, I don't like to say it, but it even outperforms my most favorite tetracycline aptamer. So our new topramycin aptamer is even better than the tetracycline aptamer. And in the meantime, the Wöhner group, they also solved the structure, both of the lower and the upper binding site. So these are preliminary data. You can see on the left side, it's a more, yeah, this is the inactive part. It's a more simple binding pocket. On the right side, you have a more peculiar folding of this binding pocket of tetracycline, of topramycin. And if you have more questions to the structure, you can go to Leon, he's in the back, and he can explain you more the details on his poster. And I think with this, I'm at the end. We have another poster with a caffeine riboswitch that we also developed or selected in a similar way. Also here we have a poster, and Vincent can present this. Yeah, and with this, I'm at the end. Short summary. I hope I could convince you that aptamers are really cool tools to engineer riboswitches and control gene expression at different RNA levels. Gave you some mechanistic insights, how these regulating aptamers work. And for us, with the capture selects, we were able to really fast find new aptamers. But to be honest, we could not find aptamers against any of our beloved targets. So there is, of course, always this chance of not finding an aptamer. And with this, I'm at the end. There are a lot of very talented PhD students in my lab. 3 of them are here in the back. And yeah, thank you for your attention.